Thank you all for being here today with us. It's a pleasure. So today I will talk about carotenoids and how to use them for cancer treatment and cancer prevention. So this project, we have been working on it since a very long time. Well, I hope you can hear me well. So in case there is a problem, please tell me, but it should be okay. So thank you, Mairim, of course, for the invitation. This is very nice from you. I was in Universidad Federal de Sergipe in September. So you can see on this photo lots of friends, lots of colleagues. So I am here with Raphaël Grunier, who is in Paris, and Mairim is here on the photo, and you can see Professor Lucindo too. So we have been working together since several years. So what I want to tell you is that my first time in Brazil was in 2015, and since that time we have a network of colleagues, of friends working all together. So we try to have some relationship between French and Brazilian researchers working on natural products. So we have built a network which is called the Franco-Brazilian Network on Natural Products, so FB2NP. And at this time, this network is trying to get some money to work together, so to invite some professor in France, and also to invite some professor in Brazil, and also so we want to increase student exchange, like a PhD student, postdoc student between France and Brazil. So I'm managing this network, and we are really very happy to work with Brazilian friends, Brazilian colleagues. So Professor Lucindo Quintans is a very good friend of mine, especially I met him in Petrolina in my first day in Brazil, and we have been developing some projects together, working on natural products like terpenes, flavonoids, and carotenoids too. And of course, we really want to increase this network to have some new friends, to have some new projects, and try to exchange some new students between France and Brazil. So before I start talking about carotenoids, I'd like to tell you that when I was in Sergipe, in São Cristóvão, in September, so I could meet all this team working at UFS, and lots of colleagues are really amazing, very nice people. So it's a pleasure to work with you. It's a pleasure to have some projects with you. And also my opinion is that we are sitting on a gold mine, because Brazil for pharmacognosy is amazing. The Brazilian medicinal plants, we really can find some interesting compounds. It's really amazing. And to me, that you have the chance to live in an amazing country, and you have the chance to work with natural products which are very interesting. So in my opinion, there are lots of possibilities for developing some new products, developing some new economy using this natural product. So I really want to increase this network and go on working with you. So after my talk, I propose that Luis could explain what he's working on and why he's in my lab now, and also explain what is important in his opinion for a Brazilian student to be able to come working in France, making a PhD or making a master training period in France. Okay, so now I'm going to talk about carotenoids. So in fact, I will give a short presentation of my lab, my team. I will talk about cancer and explain why people die from cancer, and explain how we discovered that some carotenoids are really interesting for cancer treatment. So what they look like, how we can use them for prevention or treatment of cancer. And I will show you a few experiments that we made in my lab with my student about carotenoids. Can you all hear me well, Mairim? Can you hear me?
Yes.
Okay, so it's perfect. So thank you so much.
Sometimes the internet is not so good, but it's okay.
Okay. So this is just to show you where we are exactly, because maybe you don't know in detail where is La Rochelle. So I'm going to show you change pointer. So this is Europe, and France is this small country in Europe, very small as compared to Brazil. And La Rochelle is located on the Atlantic Ocean. So we are in the middle of France, exactly here, and we are facing the Atlantic Ocean. In fact, from my lab, I just have to cross one street, and I am on the beach where there are some boats, and so we are very close to the ocean. This city is very nice, very pleasant to live, so we don't have any people are sometimes a little bit crazy. So Paris is very close. In fact, it's like I would say 300 kilometers. So going to Paris by train is very easy. As we are located close to the sea, my lab is working mostly on the sea coastline and also on marine natural products. We have a lot of projects dealing with marine natural products. So this is the university, La Rochelle Université. At this time we have around 9,000 students. So it's a very friendly university, not so big. People know each other. We have some good libraries. The students, they can make a lot of sport, and we have some very good sport equipment. And in my opinion, students are very happy to live here in La Rochelle because it's a very friendly city. We have some good restaurants, good places to go, and so student life is very, very nice. So this is my lab. The lab is called UMR CNRS Liens. So CNRS is like a label for the lab, which is French National Scientific Research Lab. So it's a quality label for the lab. And Liens means that we are mostly working on the coastline, littoral, and environment and society. So we try to find the link between the society and the coastline. And of course, for chemists, what we are interested in is looking for some natural products to find some treatment for cancer, for inflammation, and this kind of disease. So in my lab, it is very strange because we have some mix of colleagues that work like some chemists, some biochemists. Also, we have some ecologists, marine ecologists, working with dolphins or working with whales. We also have some historians, some geographists. So it's a strange lab. In fact, it's not only biochemistry; it's a mix of everything, you know. But we are all working on the coastline. Like historians are working, for example, on the migration of people, how they took the boat to go to America, and so on. So as for us as biochemists, pharmacologists, we have a lot of equipment, as you can see, for working. So we can make a high resolution mass spectrometry, NMR, microbiology, marine microbiology, extraction of natural products using microwaves. We have some pharmacology facilities, cell culture experiment. We have some chemistry lab and molecular biology labs and so on. So our project mostly deal with natural products. So in fact, we have 3 goals. The first one is to try to prevent or treat cancer and inflammation. Second one is to fight infections, so bacterial and viral infections. And the third one is about neurodegenerative disease and cardiovascular disease. So we are working mostly with some pharmacomodulation of natural products, like for example, chemists that they synthesize some kinase inhibitors. Also, we work with medicinal plants. So we are working with littoral medicinal plants, but also Brazilian or New Caledonia medicinal plants. And of course, we have a big activity on marine drugs. So we are working on seaweeds, on microalgae, on mollusk pigments. So in my team, we have different expertise, I would say. Some are working on polysaccharides, some are working on proteins, and some are working on pigments. So I'm in charge of managing a small research group which is interested in pigments and heterocycle for anti-cancer and antibacterial activity. And I love to say that this group is not French group; it's a Franco-Brazilian group, because we act with Brazilian, like for example Professor Jackson Guedes, who is in Petrolina in Pernambuco. This guy, Raimundo Gonçalves de Oliveira Jr., he was formerly my PhD student, and now he's a professor at Paris University, so he's coming from Petrolina too. He was working with Jackson in Petrolina. We have Luiz, who is a PhD student, and we have lots of works together with Brazilian colleagues. So here are a few examples of the projects that we developed this last year. So we were interested, for example, in extraction of natural products from microalgae or from medicinal plants using microwave-assisted extraction. We have purified some new pigments from microalgae and plants, and for example, we found one microalgae extract which is very interesting for treating acne and bacterial infection. So we use this pigment for phototherapy. That means that when we combine the pigments with the light, we can kill the bacteria. We have some European programs running on microalgae, and also we work on flavonoids, for example, some cytostatic flavonoids from New Caledonian and Brazilian plants. So I will talk about cancer. So the first thing that I have to say is that cancer is a disease that can be partly prevented through the diet. In fact, the food and lifestyle is very important for preventing cancer. In food, you can find a lot of chemopreventive natural products, so it depends on what you're going to eat. But if you decide to eat this kind of food, probably you will live less time than if you decide to eat this kind of healthy food. So it's like, it's like a seed. When you plant a seed in the ground, the seed is going to grow depending on the environment. If you give the seed some food and some water, it's going to grow. So it's the same. It is very well known that, for example, in Japan, most people eat some very good food, and so they don't have a lot of cancer. So first thing to say is that around half of all cancers can be prevented through diet. But of course, in some cases, when you have cancer, it could be like a genetic problem, or also you have a mutation in your gene. And in this case, you have to go to the hospital and you have to follow what I call the clinically validated protocol. So that means they will give you chemotherapy, radiotherapy, could be also immunotherapy. So at this time, some cancerologists believe that in 10 or maybe 20 years, we will not use any more chemotherapy because chemotherapy is too toxic for the people. But in fact, chemotherapy and radiotherapy can cure like around 80% of all cancer. The problem is for the 20% of the remaining cancers, because cancer cells can become chemo-resistant and they can lead to metastasis. And for this kind of cancers, we have some problem for finding a good treatment. So the question is, can we use some natural product to improve the cancer treatment? So my idea is that even if you cannot treat totally the cancer, that means maybe people will live with their cancer. You will not eliminate the cancer from the chronic disease that you can live with. That means you know you have a cancer, but you can manage it and you can live for a lot of years with your cancer. So if you have a treatment which is available, of course you should follow this treatment. So remove as early as possible the tumors, make some immunotherapy with some innovative immunotherapy strategies. And now, we are also, they are making some genetic and epigenetic profiling of tumors to identify some relevant molecular targets. So when you identify the genes that are mutated, then you can give the good drug to cure the cancer. So this strategy allows to cure around 80% of all cancer using some pro-apoptotic drugs. And in this strategy, in my opinion, natural products have a very important role to play, because you can sensitize the tumor cells to the effect of chemotherapy and radiotherapy. I just would give one example, which is very well known. For the women who have breast cancer, it is very well known that if they eat some omega-3 fatty acids, then they're going to have a better answer to the treatment. So they're going to have a better remission of the tumor, destruction of tumor cells. And so natural products can help sensitizing the tumor cells to the effect of chemotherapy or radiotherapy. And for the other cancers that belongs to the resistant cancer, resistant to pro-apoptotic drugs, the question is it really a good strategy to use some chemotherapy or use some radiotherapy? Because you know that the cells are going to become resistant. So after a few weeks, a few months, you will not be able to kill them using this. That do not rely on apoptosis of the cells. And also you can find some natural products that will target the metabolisms of cancer cells. And also find some natural products that could be interesting for killing cancer cell resistant to pro-apoptotic drugs. Like, for example, phototherapy, anti-tumor phototherapy. So, in fact, this idea of using natural product for cancer treatment, cancer prevention, was also it also arises from reading some books And this book in France is very famous. So this book is called Anti-Cancer. It was written by a very famous French neuropsychiatrist, so David Servan-Schreiber. This guy was a very famous scientist. He was working in the US. He was very active, going to a lot of congresses, making a lot of research and so on. And he didn't take a lot of time for him, for his health and so on. And one day, one of his colleagues asked him to come in his lab because he needed to make a imaging of the brain. So he asked him, could you please help me to go in this equipment for just checking that my equipment is working well? And at this time, he was diagnosed with a glioblastoma, with a brain tumor.
Professor, sorry, their presentation stopped.
Okay. I'm going to put it again. Can you see it now?
No.
No?
Not yet.
No, I'm going to try to charge it again.
Okay. Thank you.
Okay. No problem. All right. Maybe I can. Is it okay?
Yes.
Yeah. All right.
Thank you.
Okay. So this guy, when he was diagnosed with a glioblastoma, he went to see his doctor, his cancerologist, and the doctor said, "Okay, guy, he was a medicine doctor, so he asked him to tell the truth, and his doctor told him he was going to live for 6 years, and at this time he decided to totally change his food, his diet, his lifestyle." And to strictly apply an anti-cancer diet, including some fruit, vegetables, spices, cereals, low sugar intake, make some sport, make some meditation. And in fact, instead of living 6 years with this tumor, he could live 19 years, which means that just changing the food, changing the lifestyle can really have a significant impact on the development of the tumor. So during this time, he wrote this book, which is I think maybe you can find it in English. It's a book about the anti-cancer effect of food nutrients. So he talks about algae, flavonoids, carotenoids, vegetables, mushrooms, and so on. So I really recommend, if you have the opportunity to read this book, it's really a very good book. So now the question is, how did we focus on carotenoids? At this time, we were working with some microalgae, and we were trying to find some anti-cancer compounds in microalgae extract. So we were making some microalgae extract in ethanol, in water too. And every time we were trying to purify a good compound with a good anti-cancer activity, we were finding some carotenoids. So that was very strange to us. We were always finding some pigments. So lots of compounds in microalgae can have some anti-cancer activity, but a lot of time when you are looking for some anti-cancer drugs, you find some pigments like carotenoids. So we found violaxanthine, some carotenoids in Cyanophora paradoxa, which is another microalgae. So we found a lot of carotenoids from marine microalgae. We were very interested in epoxy carotenoids like fucoxanthine. I'm going to show you some example of the effect of fucoxanthine. And also we are trying now to work with some other sources like
Sorry, we are not following your presentation. We are seeing the same slide.
Okay. So I have to see why this is doing this. Sorry. I'm going to try to. Sorry for this. Écran entier. Autoriser. Is it okay?
Yes.
Yeah? All right. Sorry for that. So now we are trying to work with some other sources of carotenoids, like for example, archaic carotenoids, and also some carotenoids from plants, like medicinal or food plants. For archaic carotenoid, as you can see, this paper we wrote it in collaboration with some colleagues from University Federal of Sergipe. Lucindo is a co-author of this paper. Can you see the next slide now?
Yes.
Yes.
All right. Okay. So I will just give you a few information about carotenoids. So these carotenoids, they are lipidic pigments. They belong to the family of terpenes. So they are tetraterpenes, usually contain 40 carbon atoms or 45. And usually these are some pigments, so they are red, orange, and yellow. You can find them in a lot of producers, like some microalgae, some seaweed, some terrestrial plants produce carotenoids, some fungi also, like superior fungi, Basidiomycetes, some yeast, even in bacteria, like for example Staphylococcus aureus is producing some carotenoids, some archaea are producing carotenoids, and of course you can find them also in consumer organisms, like if you take a blood sample in humans, you will find some carotenoids. So it's a very large, very vast family, and at this time more than 1,000 natural carotenoids have been identified. And the question is, what is the biological function of these compounds, and how can we use them for cancer prevention and cancer treatment? So here are a few examples of very well-known carotenoids. So you can see their structure. So this is the lycopene that you find in tomato. This has anti-cancer activity. This is the beta-carotene that you can find, for example, in cantaloupe. I think, how do you call it in Brazil? Okay, girimon. Okay, so this is the zeaxanthine that you can find in milio, in maize. This one is a crocin that you can find in saffron. This one is beta-ionon that you can find in some violet flowers. And this one is a specific marine carotenoid, which is only found in brown seaweeds. It's called fucoxanthin. I will talk about this one because it's very interesting. So, as you can see, these compounds, their structure is quite complicated. It's not so easy for chemists to synthesize this kind of compound, especially when they contain some epoxy function, and some of them could have some alanic bonds and so on. And some glycosylation and so on. In fact, there are a lot of diversity in terms of chemistry of carotenoids. So you can find some epoxy, alcohols, ketones, lots of them. So I'm just going to show you a few examples of some what I call some strange carotenoid. So for example, you can see this one. It is an epoxy carotenoid, and it contains also an intramolecular cycle. So this kind of compound is very difficult for chemists to synthesize. So the best way to obtain it is to purify them from the biological sources. You can have some aldehyde carotenoids. Some of them are highly glycosylated, like this one. Some of them also can contain some glycosylation, and esterification with fatty acids. And as you can see, this is just to show you, some of them are really complex compounds that are not so easy to synthesize, and most of them are not even sold, as a standard. You have to purify them if you want to work with them. So I'd like to tell you that if you need some information about carotenoid, the best source is the Japanese database on carotenoid. So this database is a source of information if you want to have a structure, a biological activity of all carotenoids. So the question now is, why do all these organisms produce carotenoids? Some carotenoids are involved in photoreception, for vision, for photosynthesis, for movement in direction to go to the light, so for phototropism. And usually they are associated with some proteins. So they also play a function in stabilizing the membrane, for example, the thylakoid membrane in the chloroplast. Some of these carotenoids also, it is known that they play a role in antioxidant activity. So, for example, bacteria like Staphylococcus produce some carotenoid like staphyloxanthin because it allows them to avoid the toxic effect of reactive oxygen species. So it allows them to survive to phagocytosis and so on. Some carotenoids found in plants also play a key function in photoprotection against eye light. So when you have eye light irradiation, you could have de-epoxidation of these carotenoids to produce some derivatives, and this allows to limit the risk of burning for these plants. Some carotenoids derivatives, especially some small compounds like abscisic acid, also they play a key function as phytohormones for allelopathy, to avoid predation or to attract, for example, the bees for pollination. So some of them are chemoattractant. So, for example, damascene, which is a very good smell that you find in the rose. So they could play a role as a chemoattractant because of the smell, because of the color or the taste of the compound. And some of these compound carotenoids also play a key function in regulating the cell cycle metabolism, the cell differentiation. So, for example, retinoic acid is a very well-known carotenoid derivative, which has a lot of activity during the fetal development. So, the question now is, as we can eat a lot of these carotenoids in fruits, in vegetables, is it realistic to talk about some pharmacological activities of carotenoids? I mean, are these carotenoids assimilated by the body? Is there any absorption and metabolism of this compound? So what is known at this time is that some of them are very well transported into the blood, bound to lipoproteins. They could be concentrated, for example, in the retina or in other tissues. It is demonstrated that some of them can follow intestinal resorption using a passive or facilitated transport system. And what is very well known is that for some of them, they have in vivo activity. For example, some of them, like fucoxanthin, could induce lipolysis, have anti-tumoral effect, chemopreventive effect. So at this time in the scientific community, the discussion is not about this question. It is quite demonstrated that carotenoids can be assimilated by the body. And also some pharmacodynamic targets have been identified for carotenoids. So they could go to act on nuclear receptors, induce apoptosis, act on the cytoskeletons of cancer cells, and so on. So I'm just going to show you a few elements to have a good idea of how to use them for prevention of cancer. If you use an animal model, for example, some mouse, and you induce tumorigenesis using a chemical, then you will have 100% of mice developing some solid tumors. And if you add in their diet some carotenoids like beta-cryptoxanthine, so as you can see, the dose is 1 to 10 milligrams per kilogram each day, and then you will reduce the tumor incidence around 50%. So what was demonstrated is that in these animals, the cancer cells die, you have inhibition of metastasis, you have inhibition of angiogenesis, tumoral angiogenesis, you have some downregulation of some receptors which are involved in invasivity of the tumor cells. And it's the same if you take some animals, so you induce tumors using some 4-ball esters, which are some chemicals. And if you add to drinking water some carotenoids like pyridinine at 1 micromolar, which is a very low concentration, then you can have like a total tumor inhibition. It's just amazing. So it's really amazing to see that just adding one compound in the drinking water and you inhibit all the development of tumor. I'd like to say a few words about fucoxanthin. Fucoxanthin is carotenoid which is exclusively found in brown algae. So you can find it in seaweeds, in brown seaweeds, and also in microalgae like diatoms. This compound probably is one of the most abundant carotenoid on Earth because it represents a very big biomass, and it has a particularity of having an epoxy function. So this function probably acts as a pro-oxidant activity. So for most carotenoids, it is said that they have an antioxidant activity, but for some of them, researchers believe that they can kill the cancer cells because they have a pro-oxidative activity. So when you eat fucoxanthin, so it's not toxic at all, you can eat grams of fucoxanthin, there is no problem. It will be transformed metabolized into fucoxanthinol. And this compound has a very high activity in inhibiting tumor growth. So for example, here you can see a control mouse with some solid tumor on the skin, and the same mouse which is fed with fucoxanthin, and you can see that the tumor is cannot be seen. So it's really some researchers talk about the treasure of the sea, treasure of the sea. So it's like an amazing compound which could probably have a lot of interesting development for human health. If you work with some melanoma tumors, you can see that fucoxanthin is inhibiting the growth of the melanoma and also inhibiting the invasivity of melanoma and inhibiting the tumor angiogenesis, so the formation of blood vessels to feed the tumor. This compound, fucoxanthin, is already sold as a food supplement, so it is sold for losing some weight, but probably it could be interesting as an anti-cancer compound. And a lot of pharmacological studies have been made to understand what is the effect of this compound. So basically, it will induce apoptosis in the tumor cells. It will stimulate activity of caspases, which are enzymes involved in cell death of tumor cells. And we have shown that probably this compound is able to integrate some cell membrane and lipid raft in the cell membrane of tumor cells. So remember, fucoxanthin, probably it will be one of the compounds that will have that will be the most promising for future studies of anti-cancer carotenoids. So when you have a look to the literature, you find a lot of paper. It's like you can be drawn very easily about cancer prevention by carotenoids, anti-proliferative activity of carotenoids, anti-cancer activity of carotenoids, and so on. So now it's really very well known that these compounds have a very strong anti-cancer activity. So now we have to think about how to use them. Can we use them, for example, as food supplements? So when you eat some fruits and you eat some vegetables, I mean you have what I call the healthy food, and you have a normal intake of carotenoids. So these compounds will have some antioxidant activity and will prevent development of cancer cells. Probably also they have some effect on immune cells, on the immunity, anti-tumor immunity. And it was shown that they can activate anti-inflammatory genes, regulate apoptosis, induce apoptosis of tumor cells, and so on, and have cytostatic activity, so they limit the growth of tumor cells. So if you decide to increase the amount of carotenoids in your diet, you can have a food supplementation. So for example, you can add some beta-carotene in your diet or some fucoxanthin. And in my opinion, this should be done only for people who are under who need a treatment for cancer, so to sensitize tumor cells to chemotherapy or radiotherapy. But there is a big danger. The question is, these compounds are antioxidant, most of them, so when they go into the body, they're going to be oxidized. They're going to oxidize instead of the body lipids and body molecules. So the question is, can we use this carotenoid, for example, for people who smoke? So there are some clinical studies about this. So the hypothesis is that beta-carotene is chemopreventive, cytotoxic to tumor cells. So we could imagine that if you mix beta-carotenes for smoke, if you eat beta-carotene, you could limit the risk of oxidation and the risk of cancer, which is linked to smoking. But in fact, the data have shown that if you take a lot of carotenoids and you smoke, it could be tobacco or alcohol or pollution, there you're going to have a high amount of pro-oxidant derivatives that come from the beta-carotene. And these derivatives have pro-carcinogenic activity. So in fact, you think that you eat beta-carotene and you're going to protect yourself to prevent cancer, but in fact, if you smoke at the same time, then you will have pro-carcinogenic activity. So it's always the same, you have to be careful when you take some food supplement about the environment. If the environment is healthy, it could be interesting, but if the environment is toxic, then you can produce some toxic compound in your body. So what is recommended for smokers is not to consume some carotenoids as food supplement. So now I will talk about what we are doing in my team. So as you know, we are working with microalgae, seaweeds, plants, trying to purify, extract some natural products. So we work with carotenoids, but also with flavonoids. We have some quinone, some lots of different kind of compounds. In my lab, we have some cell culture experiment, pharmacological assays, and for in vivo assay, we work with partner labs. So this is just to show you what we are doing for identifying some carotenoids in microalgae. So in my team, we have a high resolution mass spectrometer. We can use it for identifying, separating, and identifying some carotenoids. So the idea is that we make some extract, we inject them in a UPLC system, which is associated to high resolution mass spectrometer. When this compound arrives in the mass spectrometer, they will be ionized, so we can determine their molecular weight. And at the same time, we are going to increase the energy to break them and to get some fragments, so we can determine the molecular weight of the fragments, and this allows to get the identification of this compound. So this is just an example. If you take this carotenoid and you don't break the compound, you will get a peak at this molecular weight. So as you can see, we have 4 scores under so it's a very high resolution analysis. And if we work with high energy, we break the compound, and we know exactly where the fragmentation is going to be made. And so we have validation of the chemical structure of the compound. So using this strategy, we made some database for some pigments, and with this kind of database, it's very easy for us to identify all the pigments in microalgae, or even in seaweeds or in medicinal plants. So this could be carotenoid, but also some chlorophyll derivative, flavonoids, and so on. Now we talk about one part of Raimundo's PhD. So Raimundo, he was in PhD with me from 2017 to 2020, and he was recruited as a professor in Paris in 2022. And his PhD was about sensitizing melanoma cells to chemotherapy using some flavonoids and some carotenoids extracted from Brazilian plant and microalgae. So when he arrived from Brazil, is that he knew a plant in Brazil which is called urucu, so it's Bixa aureliana. This plant, is a red plant that is used by the Indians. So I'm not going to talk about melanoma. This plant I'm going to show you is this plant that the Indian use for putting some red, on the skin. And this plant is not toxic. It is, the seeds are used for making some pigment for food, and also it's a commercial plant. It's not toxic at all. So in this plant, what was known when we started the experiment was that the red pigment was a carotenoid derivative which is called bixine. So he came from Brazil with some seeds, with some Bixa aureliana seeds, and he made some extracts using some solvents like hexane, chloroform, ethyl acetate, methanol, and all these extracts were analyzed using a high resolution mass spectrometry, and also we made some anti-proliferative assay in melanoma cells using this extract. So in the extract, we could find lots of compounds, so some flavonoids, some carotenoids, and something very interesting is that we found some new carotenoid derivatives. So in fact, Raimundo discovered 2 new carotenoid derivatives, which were unknown at this time. And he could show that these derivatives have anti-cancer activity. And also he could purify the bixin, which is a red pigment, the most abundant pigment in the extract. Also, this plant contains some tocotrienol, which are vitamin E derivatives, which are also very interesting as anti-inflammatory compound. And it also contains some flavonoids, which could be also interesting as an anti-cancer chemopreventive compound. So we focused our study on bixin. So we identified the two new carotenoids, and then we purified the Z isomer of bixin. So as you can see, it's a red pigment. It's a red apocarotenal. It's a carotenoid derivative. So we could identify the pigment using MS analysis and NMR analysis and bidimensional NMR analysis. So we were absolutely sure that it was a Z isomer of bixin. And after that, we studied the activity, the pharmacological activity of bixin. So what you can see here you have some control melanoma cells growing in a cell culture system. And when you go to increase the concentration of bixin, then you have an anti-proliferative activity. You limit the growth of these cells. As you can see, the concentration here are quite high. 20 micromolar, 50 micromolar are some high concentration when you work with some pharmacological compound. So in fact, this bixin is not so toxic. You can even eat it, it will not kill you, it's not toxic. But what is interesting is that bixin has like a cytostatic activity. It's not a cytotoxic activity killing the cells, it's more a cytostatic activity. That means that if you kill the cell, if you have apoptosis of the cell, you could have some chemoresistance. I'm going to show you that bixin, in fact, has some pro-apoptotic activities, but you have to work with very high concentration to get some apoptosis. And we identified that its activity is more like cytostatic, limiting the growth of tumor cells. Also, he made some cell migration assay, and he could show that bixin is able to limit the migration of melanoma cells. So that means that it's going to limit the invasivity of tumor cells, and it could limit the metastasis of tumor cells. And when you combine bixin to an anti-cancer drug, so this anti-cancer drug is called dacarbazine. So I'm going to show you on this figure. Here you have some melanoma cells which are treated with vemurafenib, which is a BRAF inhibitor. So BRAF is a kinase, a protein in the cell, which is often mutated in melanoma and which induces proliferation of melanoma cells. So vemurafenib is used as a drug in clinical treatment of melanoma to limit the growth of melanoma cells that have this mutation. What you can see here is that, in fact, these cells, they are sensitive to vemurafenib. So when you add some small concentration of vemurafenib, you have a high growth inhibition. So for these melanoma cells, which are sensitive to vemurafenib, even if you add some bixin, you don't have any change in the effect of vemurafenib because they are already sensitive to vemurafenib. But for dacarbazine, dacarbazine is a compound which is an alkylating agent. It's a DNA alkylating agent, which is used also for treating melanoma. But as you can see here on the blue line, when you use dacarbazine alone, you have to reach very high concentration of dacarbazine, even to get 40% of growth inhibition. So that means that these cells, these melanoma cells, they are resistant to dacarbazine. You cannot kill them using dacarbazine alone. And when you combine dacarbazine with Bixin, then you can see that you increase the growth inhibition, you increase the sensitivity. So that means that in fact, Bixin is able to sensitize the melanoma cells to the effect, to the toxic effect of dacarbazine. So this is very interesting. That means that for cancer patients, they can eat some food supplement like Bixin, which is not toxic at all. And if Bixin is going into their bloodstream and going to their tumor, Bixin could sensitize the tumor cells to the effect of anti-cancer drugs. So that's a new concept. That's the idea of people who have melanoma, metastatic melanoma. So in this case, in our cell line, in our melanoma cell line, when you use vemurafenib, these cells are sensitive to vemurafenib. Well, so even at low concentration, you have a high growth inhibition. So there's no need to add Bixin, because Bixin is not going to improve the effect of vemurafenib because they are already sensitive to vemurafenib. But for example, for dacarbazine, which is another anti-cancer drug, it's a DNA alkylating agent. You see here, even at very high concentration of dacarbazine, you cannot kill the cell. You have like 40% of growth inhibition, so 60% are surviving. And when you combine with Bixin, then you increase the sensitivity of the cell to the effect of dacarbazine. So that's a new concept, that's a new idea that we want to introduce in cancer treatment. It's not only about taking a drug against cancer. It's the idea that if you eat some food supplement like the carotenoids, you could improve the efficacy of your treatment. And this is already well known for fatty acid, omega-3 fatty acid. And what we have shown here is that in vitro, we can improve the chemosensitivity using some carotenoids, sensitivity to anti-cancer drugs. So it was the same when we studied the cell migration using a combination of anti-cancer drugs and Bixin, and we could see that combining both of them limits the invasivity of cancer cells. So in this idea, if you can eat Bixin for a patient who has cancer, it could help to limit the metastasis of tumors. And what we have shown using this, can you hear me, Mairim?
Yes.
Okay. What we have shown using some fluorescence labeling is that when you combine an anti-cancer drug with Bixin, then you are able to induce apoptosis of this cancer cell, so induce the cell death of this cancer cell. And probably this cell death is induced by the production of reactive oxygen species, because if you use a fluorescent compound that shows the production of reactive oxygen species in the melanoma cell, you can see that Bixin and the combination of anti-cancer drug dacarbazine and Bixin is inducing the production of reactive oxygen species. So that means that in fact it's like inducing the oxidation of the membranes of the cell. So we could validate all this with a cell cycle analysis, showing that the cells, the melanoma cells, are blocked before mitosis, and they will go to apoptosis. So finally, this is the conclusion of this PhD study. Raimundo, he brought some Brazilian plant. He could identify some new carotenoid derivatives from this Brazilian plant, and it could show that the bixin, the red pigment, which is not toxic, is able to sensitize the tumor cells to the effect of anti-cancer drugs. So that means that's a new idea of, improving the effect of anti-cancer drugs using some natural products which are not so toxic. So finally, I'm going to conclude. What is known is that eating some carotenoids has a high impact on your health to prevent appearance of tumor cells, to prevent the growth, invasivity, and metastasis of tumor cells. And also, as I have shown you, if you give carotenoids to animal models, you can inhibit the initiation of cancers by toxic compounds. And these carotenoids, some of them have direct cytotoxicity on tumor cells or cytostatic activity. They can inhibit tumor angiogenesis, so formation of blood vessels around the tumors. They can sensitize the tumor cells to the effect of anti-cancer drugs. And also they could be used as possible photosynthesizers to kill cancer cells. And what is not known at this time is that if these carotenoids could be useful for improving the efficiency of radiotherapy of tumor and also modulate the immunity of tumor. So these questions are not really discussed in the scientific literature. So it could be interesting to study the effect of carotenoids in immune cells. So I want to just to conclude. Thank you again, Mairim, for this kind invitation. Working with you and knowing you is a real pleasure. I want to thank all the founders of this work and all researchers involved in this project. So some of them are Brazilian colleagues, which are involved in the Franco-Brazilian network on natural products. And we have a special thanks for University Paris Descartes with our colleague Raphaël Grunier and Raimundo Gonçalves. So we work a lot with them on natural products. Thank you for your attention. Any question?
Thank you, Professor. Pessoal, vocês têm alguma pergunta? Eu vi que Cíntia fez uma pergunta.
Okay. Okay.
Any student made a question. I am trying to find. If you have articles on a specific use as a protector for smokers. I guess she is trying to ask if the carotenoids can be used to protector for smokers. In this case.
Because if you smoke and you eat a lot of carotene, for example, then you're going to oxidize the carotene. And so this is a clinical study that was made in France. I can find some information and send it to you, send it to Mairim. But what they concluded is that for smokers, it's better not to eat some food supplements with carotenoids. In fact, it is, I would say, counterintuitive because these compounds, they have like antioxidant activity. We could think that they have anti-carcinogenic activity, but if you smoke, and then you're going to induce the production of toxic compound from carotenoids. So at this time, it's not recommended to eat some carotenoids using for smoking.
Ok, thank you. Cíntia, tudo ok? Acho que travou de novo.
Yeah.
Hello?
We're back.
Yeah, we are back.
Yes,
Okay.
thank you. I have a question. When you extract these carotenoids, is in a lower amount. It's a lower amount of substance. Yes. Because
Yeah.
when we extract the essential oil, for example, it's very lower. Do you guys think about to put these carotenoids, this substance, in a pharmaceutical form, or you only test the free carotenoids?
It's a good question because for some carotenoids, they are highly apolar, hydrophobic. So it's not so easy to test them in cell culture medium. When you put them, you dilute them in cell culture medium, sometimes you see some crystals. So that means that they are not very well solubilized. So of course, when you give them to eat to animals, they are going to be included they're going to be digested and included in some kind of micron and so on. But when you work with in vitro cell culture model, you have a lot of water, so it's not so easy to dilute them. So we have already worked on making some liposomes with beta carotene to increase their effect in cell culture models. Also, we work with cyclodextrin to include them in cyclodextrin. And also we can make nano encapsulation, for example, with chitosan nanoparticle or things like this. So we are also working on it, but for some of them are quite polar and easy to work. Like flucloxantine is not so complicated to and we use some small concentration, so we can dilute them, and we manage to work with some of them. But you're right; they are not so easy to purify at high level, and also to use in cell culture experiment.
Okay. And about the patent process, when you extract this substance, you can and made the biological activities. Then how occurs the patent process there actually in France?
You mean if you want to patent the activity or the extraction?
The both, I guess, because when you extract,
Yeah.
you also can patent. Yes.
Yeah. So I would say that for these carotenoids, usually we mostly publish the data. We don't make patents, but if we could find some, well, we have developed some extraction system using microwaves and so on. So if you patent here, you have to patent before publishing, of course, if you want to protect your discovery. It's the same for, you can patent the activity in animal model or in cell culture model. And after that, when the patent is deposited in France, usually you go to internationalization of the patent. And if it's really interesting, try to find the companies that could be interested in developing this product or this extraction. In fact, we have another microalgae extract that we have patented recently, so we know exactly how to do it. But the question is, after that, you have to go to see a company to put a product on the market, or to patent the extraction, and to have the use of your patent to extract the compound. So in France, it's quite easy to do it. It can take a long time to write the patent, because it's not so easy to write the patent. And after that, you have to work with a valorization cabinet for patenting the extract. Or at this time, there are not so many patents on carotenoids, especially for food supplements and so on, because there are not so many carotenoids are extracted. Beta carotene can be synthesized by chemists. Astaxanthin is a very well-known carotenoid also, which is purified from marine organisms. But for extraction purification, usually these are some what I would say classical extraction using
Okay.
solvents and so on.
Ok, thank you so much. Pessoal, vocês têm alguma dúvida?
I have a question. Are you hearing me? Can you listen?
Yeah.
In the beginning of your presentation, you spoke about a patent for acne treatment, I guess. Am I right?
Yeah. Mm-hmm.
Is it related to the reduced inflammation or more about the antimicrobial effect of these components?
Okay, I'm going to explain. So we work with some microalgae extract. So in fact, we wanted to find some phototoxic extracts, so some pigments in microalgae extract that you could activate with light to produce some reactive oxygen species. So we grew a lot of microalgae species. We made some extract using ethanol, using green chemistry, so eco-friendly extract. And we screened them for reactive oxygen species under photo irradiation with light. And we found one, which is a diatom ethanol extract, which is highly phototoxic. So this extract, when you put some white or red light on it, it releases a high amount of reactive oxygen species. And at the first time, our idea was to use this extract for killing cancer cells. And we are still working on this project. But we also thought that it could be interesting to try to kill some bacteria, for example, skin bacteria. And then we had the idea of treating acne for dermocosmetic treatment of acne. So it's very easy, you just make the microalgae extract, you put it in a gel, in a cream, like dermocosmetic system, and with the light, it's going to activate the pigment and kill the bacteria. And this was so interesting. We made some experiment with bacteria, and the results were amazing, really, really good. So we have written a patent for using this diatom pigment extract for phototoxicity, antibacterial phototoxicity. But in this extract, the active compounds are not carotenoids. They contain some carotenoids, but the active compounds are mostly some, I would say, chlorophyll derivatives, especially theophobide A, which is a phototoxic chlorophyll derivative. So just to tell you the end of the story, at this time, we are working with a company which is making the clinical assay for developing this dermocosmetic cream, and we really hope that it's going to go to the market. So it could be very, very innovative treatment of acne using microalgae extract.
Just connecting my question. Do you take into consideration the sun exposure when developing your cream, your gel?
Yes, in fact, we studied the irradiation, photo activation with some sunlight activation, and also, using the cell phone. So you can imagine some young people with acne, and so they're going to put the lamp, and just treat like this, So we studied the white light, red light, the cell phone light, and also the sunlight. So, for treating acne at this time, you have to go to see a doctor. He's going to give you some antibiotics or some retinoic acid, which has some side effect, toxic side effect. Usually it induces irritation and so on. And our extract is the natural microalgae extract, and it has no side effect. It doesn't induce a side effect. So we hope that it could lead to a new product on the market for dermocosmetic management of acne.
Thank you.
We have another question from Pamela. She's asking about the use of carotenoids for anti-cancer activity. Are there already clinical studies with humans?
Yeah. There are some clinical studies with humans, especially with beta carotene, to cure some. I think it's mostly melanoma, and also with fucoxanthin, the Mairim carotenoid. And at this time, I don't really know if the activity has been validated in clinical assays. I'm not so sure. But for sure, in preclinical assays in animal models, it is totally validated, but not in clinical assays at this time. So at this time, at least in France, these carotenoids are not sold as anti-cancer drugs, because when you go to sell an anti-cancer drugs, you have to check a lot of things like lack of toxicity, and you have to prove using a cohort of patients. You have to prove that the effect is significant and so on.
Ok, thank you. Vocês têm mais alguma pergunta? A Renata levantou a mão.
Hi, Professor. Congratulations for your work. It's really beautiful. Do you have any study specifically with breast cancer? I'm breast surgeon. Because of that,
We some years ago we were working with breast cancer cell lines, and we could find some good activity with carotenoids inhibiting the proliferation of breast cancer cell lines. So we were working with MCF7 and MDA-MB231, and now at this time we are only working with melanoma cells. So we don't work anymore with breast cancer cells, but it's only in vitro assay, not in vivo assay for carotenoids. So the real question is why these compounds, which are so active and which are not toxic, why they are not on the market, as anti-cancer drugs or supplements. Because, for example, fucoxanthin, it's very easy to purify. Beta carotene is very easy to purify, to obtain some. These compounds, for the simplest one, are not so expensive to produce. So probably there is no interest of the scientific community to develop this kind of natural product. Because these products are not so interesting for making money,
Professor, I appreciate so much your presentation, and I have a question about your work. To know the reason why the compound is action against the cancer or another tumor, do you test for one methodology or another test to know the reason why that compound is active against the cancer or the tumor?
You mean for understanding the pharmacological activity?
Yes.
Understanding the mechanism, the mode of action of this compound. Yeah. First, when you work with anti-cancer drugs, you have to find some good control, because usually if you work, for example, with melanoma cells, the control you're going to say, try to work with some control cells. But control cells are not growing as fast as cancer cells. So, in my opinion, it's better to compare one natural product like carotenoid with some anti-cancer drugs to have a good idea of the activity of the compound. But if you compare with a cell line that grows very slowly, probably you will find that it has no effect on the normal cell line and a very strong anti-proliferative effect in cancer cell line. But in fact, it could just be explained by the fact that the cancer cells are growing very fast. So you will have a big inhibition of proliferation in cancer cells. And to understand how these compounds act in the cells, usually what you see in the papers is that people are going to make some apoptosis assay and so on. So they show that the compound is going to kill the cells, okay? But apoptosis is like a conclusion of what happened before. So the question is, these compounds, they are highly hydrophobic. So what is the most logical is that they go into the cell membrane, into the tumor cell membrane. And they could interfere with some receptors in the cell membrane. They could induce, for example, peroxidation of membrane lipids. They could induce damaging in the lipid raft in the cell membrane. So probably they don't have a specific pharmacological target. You know what I mean? They are not acting by blocking a specific enzyme or blocking a specific pharmacological receptor. Most probably these compounds, I think they have a non-specific activity. They go into the membrane, into the lipids, and they will damage the lipid membrane. So it's more like a non-specific activity, in fact.
Okay, thanks.
Laurent, thank you so much again. It was amazing. Thank you.
It's a pleasure. I'm very sorry for connection trouble because I was talking and I didn't check that the connection was still good. I talked so much, so now probably it's time for me to give the talk to Luiz.
To Luiz, okay.
He's
Thank you. No worries about the connection.
With you in Brazilian.
Yes.
Thank you so much for being here, and it's a pleasure. I really hope that we will meet very soon for true in France or in Brazil.
Are you coming to Petrolina?
I will be in Brazil in August and September in Petrolina. Yeah.
The symposium.
Yeah.
Maybe
in
you'll
September
be there.
in Petrolina. So if you go there, we could meet in Petrolina.
Maybe you'll be there. Thank you.
I hope so.
Bye, thank you. Congelou de novo. Luiz.
Isso que eu tô com saudade.
Tudo bem? Luiz, falhou ali como... Eu só entendi a saudade, não foi?
Então, eu vou falar em português que eu tô com saudade.
Ah, tá. Com certeza, fica à vontade.
É melhor. Então, eu queria compartilhar um pouco, né, da minha experiência aqui na França.
Só um pouquinho. Vai apresentar?
Não, só vou falar um pouco.
Tá, então pode fechar a tela se quiser, porque tem uma tela ali de projeção.
Pronto.
Tá, beleza.
Melhorou? Então, tem alunos de mestrado e doutorado, é isso?
Isso. De dois programas, da Ciências da Saúde e Ciências Farmacêuticas. E não, tem mais outros programas aí também. Mas a maioria é de Ciências Farmacêuticas.
Que bom, sou farmacêutico também. Eu queria compartilhar com vocês uma coisa que eu gostei muito a respeito dessa turma aqui, é que o pessoal não teve a menor vergonha de fazer as perguntas em inglês. E eu estava conversando com o Laurent mais cedo, que o que realmente destravou a minha, digamos assim, que me deu oportunidades de tanto ir para os Estados Unidos, por exemplo, quando eu passei um ano e vim para cá, foi o inglês. Então, assim, o maior conselho que eu posso dar, né, para os estudantes de pós-graduação que aí no Brasil desejam ter alguma oportunidade fora do país, é que invistam no inglês. E quando eu digo invistam, não estou dizendo que é para você pagar um curso caro, que não é para... não tem nada a ver, né? Eu mesmo nunca fiz nenhum curso de inglês de nenhuma natureza. Então, assim, foi aos trancos e barrancos, mas fui me desafiando e aceitando os novos desafios e me desafiando de novo, e assim eu consegui progredir. Estava até comentando com o Laurent que, às vezes, os alunos, meus colegas, por exemplo, lá da Univasf, eles veem assim, você como se você fosse um cara excepcional. Mas eu disse a ele, eu sou um cara extremamente comum, eu sou um cara normal, certo? Não tem nada de especial a meu respeito, a não ser encarar desafios. E acho que aqui todo mundo está encarando um certo desafio. Com seus temas de pesquisa, com seus perrengues, que faltam as coisas sempre, né, nos laboratórios, vocês não sabem. Mas o maior conselho que realmente eu posso dar é esse. Eu tive a sorte de, em 2014, né, tinha um extinto programa, não sei se vocês já ouviram falar, mas ouviram falar do Ciência Sem Fronteiras já? Acredito que sim. Né, infelizmente, né, acabou, espero que um dia volte, mas eu fui com muito medo. Mas eu fui com muito medo, sabe? Nunca tinha saído de casa, e aí decidi e falei, não, essa oportunidade eu vou ter que agarrar. Então, foi lá onde eu consegui aprimorar a língua, né, que não foi fácil, mas, de novo, você tem que se propor a algum desafio para poder progredir. E foi, inclusive, o que me gabaritou para vir para cá, né, porque, por exemplo, existia a vaga, mas não existia um estudante que conseguisse se comunicar de maneira eficiente em outra língua, que não português. Afinal de contas, só nós mesmos, os portugueses, e mais uns países pingados ali da África que falamos português. Então, quando eu cheguei aqui na França, eu só falava inglês com o Laurent, né, o tempo inteiro. E aí, com o tempo, a gente comecei a estudar francês e tal. Estou melhorando, mas ainda não é lá aquelas coisas. Né? Quando eu tenho dificuldade, por exemplo, de explicar alguma coisa a ele em francês, eu já mudo logo para o inglês, porque para mim é mais tranquilo. Falamos um pouco, sim. E é isso. A mensagem que eu queria deixar para os estudantes de pós-graduação é que é possível, sabe? E não tem nada que me faça uma pessoa diferente de vocês, não. Absolutamente. Eu vejo muitos dos colegas tendo essa impressão que não acho que reproduza muito bem o que é, sabe? Então, invistam mesmo, assim. Tentem se aprimorar. Eu comentei até com o Arthur, fiquei esperando as perguntas em português, com é porque o nome tá Mairim também, eu esqueci teu nome, desculpa. É a
Ana Maria.
Ana Maria. Eu pensei, né? A Ana Maria vai ser a mediadora das perguntas, mas não, o pessoal foi e perguntou em inglês, e isso é muito bom, isso é muito bom mesmo para o programa, para que novas portas se abram para vocês, porque é aquela coisa, né? A língua oficial da ciência é justamente o inglês. Então, todos os artigos de maior fator de impacto, as revistas, as comunicações são todas em inglês. Parece uma língua difícil, mas é porque vocês não estudaram francês ainda, eu acho. Né? E o português também é muito complicado em comparação ao inglês. Então, é muito esse mito de que inglês é difícil, mas, na verdade, é mais a gente se achar capaz do que qualquer outra coisa. E, bom, acho que o maior conselho que eu posso dar é esse. Né? Eu vim para cá por uma oportunidade do professor Jackson, né, que tinha comentado com um professor lá do meu laboratório lá no Brasil, no Laboratório de Farmacologia Experimental, lá da Univasf, que ele estava sem aluno para mandar para cá, para o doutorado. Nenhum aluno numa universidade com, sei lá, quantos mil estudantes, que conseguisse se comunicar, pelo menos, né, com o orientador e tal. E eu disse, olha, eu vou. E aí o Jackson fez a ponte, né, entre eu e o Laurent, e a gente começou a conversar, eu passei por uma audição, e aí, felizmente, consegui vir. E foi isso que me ajudou bastante. Lógico, eu tive um percurso na carreira acadêmica regular, nunca fui um aluno nota 10, sabe? mas também fazia meus deveres de casa, digamos assim, mas foi o inglês realmente que me deu a chance de ir em lugares que eu nunca imaginei, né? Por exemplo, estar aqui hoje, nem nos meus melhores sonhos eu consegui imaginar. Então, se vocês tiverem essa oportunidade, realmente vale a pena. Vale a pena. Se vocês quiserem seguir carreira acadêmica, vale muito a pena. E assim, se quiserem me fazer alguma pergunta sobre como é a vida aqui na França, como eu estou me adaptando, estejam 100% à vontade. Estou aqui para tentar passar um pouco dessa experiência.
Luiz, eu queria agradecer também a oportunidade de compartilhar, que ontem eu falei um pouco, eu fui apresentar a disciplina e falei um pouco do pós-doutorado, eles tiveram muita dúvida, né, como que é a questão da bolsa, a questão de como que faz o contato com alguém de fora. Então, tudo isso que tu falou é super importante, e a questão da língua também. E aí eu tenho uma pergunta meio besta, assim. O francês, né, o pessoal da França não gosta muito quando a gente chega na França falando inglês.
Sim.
Na universidade, todo mundo fala, não tem disciplina aí, né? Aí só vai e faz a pesquisa, né? Então,
tem disciplina, tem algumas disciplinas. Eu tenho que fazer 6 disciplinas em 3 anos. Não é muito não, mas tem.
Mas as disciplinas são em inglês ou francês?
Então, eu estou fazendo francês, por exemplo, como uma disciplina do doutorado, entendeu?
Uhum.
Mas assim, é bem mais... eu diria que as disciplinas que a gente tem que pagar aqui, elas são bem mais brandas e bem mais... são muitos temas que não necessariamente têm a ver com a pesquisa em si, sabe? Ou com a técnica que você está aplicando ali no seu laboratório. Né? São o que eles chamam de formações transversais. Então, elas passeiam por várias áreas de conhecimento. Ética, língua estrangeira, língua inglês aplicado à pesquisa científica, enfim, são muitas mais nessa linha. Um pouco diferente no Brasil, mas tem.
Mas quando você chegou na universidade, tipo, os técnicos, essas pessoas que você tem que conversar diariamente, eles falam em inglês também, só francês?
Então, alguns poucos falam bem inglês, né? A maioria falam francês. Mas assim, aqui em La Rochelle, é porque quando eu vi, eu também tinha essa impressão, né? Eu pesquisei bastante coisa sobre a França para não chegar aqui também feito um tabaréu sem entender nada. Então, eu justamente vi essa questão, né, da língua e de não chegar falando inglês, já que eles não gostavam. Mas eu tive a impressão que isso acontece mais em cidades grandes, né? Acho que em Paris eles são um pouco mais fechados quanto a isso, sabe? Se sentem às vezes até um pouco desrespeitados se você já chega falando inglês, né? Tem que você dê um bonjour primeiro para depois começar a falar inglês. Mas aqui em La Rochelle não. É uma cidade que respira a universidade, né? É uma cidade com uns 75 mil habitantes e 9 mil são estudantes. Então, assim, é uma proporção muito grande de estudantes, né? Então, tem gente do mundo todo. É uma universidade que acolhe muitos estudantes de outros países. Então, assim, entre os estudantes, o inglês, lógico, tem alguns que dominam mais, outros dominam menos, mas sempre, por exemplo, na vida cotidiana, com os colegas doutorandos, né? A gente sai, a gente vai num bar, tomar uma cerveja. Quando eu tenho dificuldade mesmo em me comunicar em francês, eu troco para o inglês e é super tranquilo. Então, assim, eu acho que depende muito da cidade, como é a vida na cidade, né? Aqui em La Rochelle, como é uma cidade tão grande, uma cidade ótima, maravilhosa, linda, e as pessoas são bem mais receptivas do que nas cidades maiores. Então, acho que essa questão de não falar o inglês e tal, acho que é mais importante talvez em Paris, em Marseille, em outras cidades maiores, né? Mas aqui é super tranquilo. Mas isso também não me exime da responsabilidade de aprender a língua falada aqui. Eu acho isso muito importante também, porque tem determinados... Por exemplo, você vai ter que ir ao banco, às vezes, né? Você vai ter que, sei lá, mandar alguma correspondência, enfim, na vida cotidiana mesmo, pede algumas... E aí, se você se confiar 100% no inglês, às vezes você vai ficar na mão. Então, assim, ao passo que eu tento melhorar o francês, eu deixo o inglês ali como um coringa, que às vezes quando não dá, quando a comunicação está complicada, aí é mais um recurso.
Ótimo, obrigada. Pessoal?
Uma dúvida só. Há quanto tempo O senhor está em Paris.
O senhor está no céu, rapaz.
Há quanto tempo você está aí na França já?
Eu cheguei dia 5 de janeiro do ano passado. Então tem pouco mais de um ano. 1 ano e 2 meses, aí, quase 3. Que eu cheguei, assim, tô amando mesmo. La Rochelle é uma cidade maravilhosa. Alguém mais?
Eu aqui.
Oi,
Você teve um tempo assim de preparação do seu currículo para poder aplicar para fora? Ou foi algo assim mais de supetão que surgiu?
Tive, e ainda assim, quando surgiu a oportunidade, acho que era meados de março de 2021. A gente ainda não tinha, eu não tinha nem me vacinado ainda. Então, assim, porque não tinha vacina mesmo, né? Tava aquela questão de prioridade de vacina, quem se vacina primeiro, quem se vacina depois. E esse era o grande empecilho no momento, né? Então, assim, eu fui preparando o dossiê com meu currículo, né? As publicações, lista de publicações, fiz um currículo apresentável ali, né? Organizado e tal, bem resumido, eu diria. E passei pela audição com o Laurent e com a outra professora, Valérie, e a gente ficou, saiu o resultado, né? Eu fui finalmente selecionado, só que eu não tinha tomado vacina e eu não tinha tirado visto ainda, estava sem vacina eu não podia viajar, e aí, enfim, ficou nessa enrolação. Era para eu ter chegado aqui em setembro de 2021, mas eu só fui chegar em janeiro de 2022. Demorou muito tempo por conta de questão de vacina e isso tudo. Com relação à aplicação, né, preparar o dossiê, né, eles chamam aqui de dossiê, mas é todas as suas informações que você tem, tudo que eles pedem, né. Tem programas que são um pouco mais complicados, eles pedem muito detalhe. Tem programa, por exemplo, aqui na França, ou na Alemanha, ou em alguns outros países aqui da Europa, que pedem todas as suas notas da graduação, todas as suas notas do mestrado, com tradução juramentada. Então, assim, é uma montanha de papel, às vezes, que você tem que providenciar, mas isso varia muito de lugar para lugar. Então, assim, por exemplo, a minha esposa agora, ela defendeu o mestrado tem um ano não, um mês, lá na Univas, e ela está tentando aplicar para vagas de doutorado aqui na França, né? E aí é bem isso que eu disse. Tem vaga que a aplicação ela é um pouco mais resumida, eles não perguntam tantos detalhes assim, talvez deixem isso para um momento da entrevista, né, da audição. E outras formas, né, outros editais, né, seriam os nossos editais aí, que são bem mais detalhados. Então, cada instituição, cada órgão que fomenta também tem determinadas regras, né? Tem editais, por exemplo, que não permitem estrangeiros, tem editais que são para pessoas até 25 anos, então, assim, tem uma grande variedade mesmo, né? Então, a depender de cada caso, você vai organizar o seu dossiê, a sua aplicação com aqueles pré-requisitos, né, pedidos lá. Mas isso varia muito também, né? Tenho visto com a minha esposa, por exemplo, que alguns processos são bem detalhados e bem maçantes, assim, muita informação que eles pedem, outros não, outros já são bem mais tranquilos. Aí vai depender.
Obrigada.
Beleza? Digam mais.
Você quer explicar o seu trabalho?
Sim. Não, ninguém perguntou, mas eu vou explicar o que eu vim fazer aqui, né? Porque ninguém perguntou, mas eu vou tentar dar uma resumida do que eu vim fazer aqui. Como vocês sabem, né, o Laurent trabalha com melanoma já há bastante tempo. Trabalhou com o Raimundo, né, durante 3 anos aí. E eu vim para cá também fazer pesquisa com melanoma. Só que a gente vai usar uma abordagem diferente para purificar compostos com atividade antimelanoma, que é o chamado de ligand fishing, que é pesca de ligante, né? Basicamente, a gente comprou esferas magnéticas, né? São microesferas da ordem de 1 micrômetro de diâmetro, que elas são funcionalizadas, elas têm um grupo funcional lá que é bastante reativo na presença de aminas primárias, que tem nas proteínas de monte. Então, através desse sistema, a gente consegue imobilizar enzimas purificadas nessas esferas magnéticas. E aí, o que é que acontece? Através desse sistema, por exemplo, um dos alvos que a gente vai usar, né, relacionado, que a gente pode usar, inclusive, relacionado à atividade anticâncer, é a tubulina. Então, imagina, a gente tem a esfera magnética, imobiliza a tubulina ali naquela esfera, e aí, a partir dessa partícula, né, funcionalizada com uma proteína alvo, a gente pode pescar ligantes, ou seja, colocar esse sistema em contato com misturas complexas, como um extrato de uma planta, uma fração que você já achou uma atividade bacana, para que os compostos ali presentes, com afinidade pelo alvo, eles possam se ligar, e a gente separa tudo isso com um ímã. É um tipo de cromatografia por afinidade. É bem interessante, mas também bem desafiador, porque eu nunca trabalhei com isso na minha vida, e né, tem algumas etapas que são bem críticas durante o processo. Então, a gente vai está tentando elaborar esse sistema de pescar ligantes, né, uma cromatografia um pouco mais refinada, mais específica também, porque não é sílica, né, não é por polaridade, é por afinidade pelo alvo, né, e também pretendemos fazer isso para alvos antivirais, né, coronavírus. Né, a gente vai utilizar a protease do coronavírus, a 3CL, imobilizada nessas partículas magnéticas para poder extrair de plantas brasileiras, de extratos de algas marinhas, de microalgas também, compostos interessantes que possam ter atividade tanto antiviral como antimelanoma. Então, é basicamente esse o resumo da ópera que a gente está tentando desenvolver aqui. Você trabalha com meu... Não. No Brasil, eu trabalhava com músculo liso primeiro, né? Trabalhei com farmacologia do músculo liso, atividade relaxante de traqueia, útero, ilho e aorta. É uma área que até o professor Lucindo, ele conhece muito bem, a professora Mairim, eu não conheço muito bem ainda a área de expertise, mas eu sei que o Lucindo já trabalhou muito com isso. Depois eu mudei para comportamento animal, completamente diferente, né? Avalia efeito antidepressivo e ansiolítico de extrato de planta em modelos de animais, né? O modelo murino, com camundongos. Isso foi o meu mestrado, inclusive, né? Foi avaliar o potencial antidepressivo, na verdade não é antidepressivo, né? Semelhante ao antidepressivo em animais de laboratório. E agora, no doutorado, mudei de tema mais uma vez, né? Quer dizer, não bastasse o desafio da língua, o desafio de estar longe da família e tal, existe o desafio também de ser uma nova temática, né, na minha carreira. É tudo muito novo, eu nunca tinha trabalhado com cultura de célula, né, tive que passar por treinamentos aqui para aprender a manejar as células e tudo mais. Ainda estou aprendendo muita coisa, obviamente, mas basicamente eu mudei de áreas já algumas vezes. Deixa eu ver isso. Sara Fernanda pergunta: no seu grupo de estudo, algum colaborador que trabalha com componentes naturais para tratar hipercromias? Acho que essa pergunta foi para o Laurent, talvez.
Hipercromias?
Uhum.
Skin da pele?
Uhum.
Penso que não trabalho. Temos moléculas que estão inibidor de tirosinas, temos, mas porque também está importante para o melanoma.
Sim.
Mas não, aqui não trabalhamos com hipercromas.
Tirosinase, era justamente tirosinase. Foi aqui. Bom, por enquanto não, Sara. Aqui realmente o foco é o melanoma. Tudo bem que a tirosinase é um alvo bem importante, mas aqui por enquanto não.
Podemos trabalhar também com tirosinase como para ligand fishes.
É um alvo bem comum, inclusive. Para a revisão que a gente fez, era o mais usado. De nada, Sara. Alguém mais? Bom, acho que não.
Just, I would like, quero dizer umas palavras português. Meu português é muito malo, não posso falar muito mais. Vocês, em Brasil, vocês têm uma oportunidade importante para trabalhar com produtos naturais. Penso que as plantas medicinais de Brasil são uma fonte incrível de moléculas muito interessantes para câncer, cancro, para inflamação, para tem livros de botanistas franceses que foi a Brasil para estudar as plantas medicinais, e esse livro ele chama Auguste de Saint-Hilaire. É um botanista francês que foi a Brasil. Tem descrição de plantas medicinais que estavam utilizadas para medicina tradicional no Brasil no século XVIII. Isso é uma fonte de conhecimento muito importante para descobrir, para trabalhar com plantas medicinais no Brasil. Porque você tem uma possibilidade incrível de trabalhar com moléculas muito interessantes. Penso que se eu sou um estúdio brasileiro, vou a ler esse livro para eleger a planta que quero trabalhar. Sim.
Sim, é muito legal isso, como as pessoas de fora veem a biodiversidade brasileira, né? A gente tenta se preocupar com a importância disso, mas, ao mesmo tempo, de modo geral, no senso comum, as pessoas não se importam muito. E vocês veem, né? É um tesouro. O Laurent bem fala isso todos os dias, que a gente tem, em termos de biodiversidade, um tesouro nas nossas mãos, né? Que se a gente tivesse um pouco mais de incentivo, né? Eu sei que vocês são muito empenhados, né? Todos os estudantes de pós-graduação, principalmente no Nordeste, a gente trabalha muito com essa parte de produtos naturais, né? Isso é uma fonte esgotável, não é inesgotável, né? Mas que precisa ser... A gente precisa ter orgulho disso, sabe? Se a gente se visse de fora um pouco, enquanto país, enquanto comunidade acadêmica, e como isso é importante para as outras pessoas, acho que a gente teria muito mais orgulho disso. Então, assim, realmente esses outros olhos do mundo todo para o país, para o Brasil em termos de biodiversidade, são realmente muito importantes, e acho que a gente deve ter muito orgulho e ao mesmo tempo se empenhar bastante para produzir ciência nesse sentido.
Quero dizer também, penso que tem uma oportunidade para a economia do Brasil, para utilizar essas plantas medicinais para fazer extratos, para fazer médicos, sim, produtos naturais, também extratos para frutas, extratos. Tem muito oportunidade.
Um potencial econômico enorme também, né? Então, assim, com essa questão de patente, né? A gente pede muita coisa, inclusive, em termos de patente, para o exterior, às vezes, porque falta incentivo lá, né? Então, acho que eu, quando saí do Brasil, você acaba tendo muito mais orgulho de ser brasileiro, sabia? Porque você vê como as pessoas veem o seu país e como elas te veem, você fala, meu Deus, não acredito. Eu não acredito que... Sabe, às vezes a gente tem um pouco dessa síndrome do vira-lata, mas não é para ter, não, de verdade. Não é para ter, não. São motivos de muito orgulho mesmo. Assim. E sair um pouco, dar essa impressão de que realmente nós estamos no centro dessa discussão. E nós lançamos tendência nisso. Né? Então, realmente, esse é um recado bem importante nessa área da gente, né? Produtos naturais.
Alguém tem alguma outra pergunta, pessoal? Não? Então, a professora teve que sair, ela pediu para eu agradecer. I would
Tudo
like
bem.
to thank you on behalf of the Professor Mairim. E
Thank you, muito prazer.
Thank you very much. Eu acho que hoje é só isso, né? Acho que we finish for today.
Ok.
Thank you.
Thank you so much.
Thank you so
I will
much.
send the PowerPoint to Professor Mairim, okay?
Okay, thank you. We'd like to
Okay.
think about this.
Thank you.
Bye
Bye
bye.
bye. See you. Bye bye.
Tchau.
Obrigado.
Tchau.
Tchau,
Tchau,
valeu.
tchau.